Let's have a small introduction to 3-point mapping. Now in the figure here we see there are two chromosomes, each having three genes at different positions or loci. Crossing over takes place between these two chromosomes, and these segments or genes may be exchanged. Now through 3-point mapping or 3-point test cross, we are able to know three things. First is the relative distance between different genes. Here it might be between X and Y, or Y or Z, or Z and X. Second is the order or sequence of these genes. Here on a single chromosomes, these three genes might be present as X, Y and Z, X, Z and Y, and Y, X and Z. No matter from which side we see the sequence, whether left or right, the point to be noted here is that in each of these three cases, the gene lying in between changes. In the first case, there is Y, in second there is Z, and the third one is X gene. The third thing that we are able to know from a 3-point test cross is the frequency or number of times crossing over takes place. Coming to the criteria for successfully mapping across, the first one is the genotype of the parent producing the gametes is to be heterozygous at all the loci in consideration. Second is constructing a cross where the genotype can be correctly determined by observing the phenotype easily. That is, we are able to observe the characteristics of the offspring or the progeny without any difficulty. And the last criteria is the size of the sample, that is, the number of offspring or the progeny should be large in number. Moving further, an important point to be noted here is that the frequency of recombination is directly proportional to the distance between two genes. That is, more the distance between any two genes, more is the likelihood that crossing over will take place, and less the distance, or if the two genes lie besides each other, the probability of crossing over taking place declines. To understand 3-point mapping, let's take an example of a chromosome having three genes A, B and C. To find out the order or the sequence in which these three genes lie, we'll have to find out which gene is present in the middle position, whether it is B, C or A. Now 3-point test cross involves crossing a heterozygous capital ABC and small abc parent with a homozygous recessive individual having constitution small abc in both the chromosomes. We will first look at the most important aspect, that is the gametes produced by the heterozygous parent. As we can see, the gametes produced by heterozygous parent can be divided into four categories. First, where no crossing over takes place. Second, a single crossover takes place in the upper portion of the chromosome. Third, a single crossover takes place in the lower portion of the chromosome. And the fourth one, that is double crossing over takes place between two chromosomes. In the first case, we will get gametes as it is, that is capital ABC and small abc. In the second case, single crossing over takes place, resulting in exchange between the second and third gene. The first one remains as it is, producing gametes capital A, small b, small c, small a, capital B, capital C. In the third case, where the crossing over takes place in the lower half of the chromosomes, resulting in gametes capital A, capital B, small c, and small a, small b, capital C. In the fourth case, double crossing over takes place. Recombination occurs in both the upper region and the lower region of the chromosome. As we can see here, this results in exchange of only the second gene between the two chromosomes, resulting in gametes capital A, small b, capital C, and small a, capital B, small c. As we can now understand that in a homozygous recessive individual, both the chromosomes are similar. Hence, even if the crossing over takes place, the genetic constitution of the chromosomes will remain same, whether a single crossover takes place or double crossover takes place. The gametes will always be small a, small b and small c. Thus, while observing the progeny of this cross, the phenotype will always be the genetic constitution of the gametes produced by the heterozygous parent. That is, whatever phenotypic character we observe in the progeny of this cross will be exactly similar to the genotype of the gametes produced by the heterozygous parent. Let's see the hypothetical frequencies observed in the cross. Here we have the genotype of the progeny or the offspring, the expressed phenotype, and the supposed frequency or the number of individuals observed. We'll assign a small alphabet to it. So the first case, in the non-crossovers, here we can see this is the progeny having one gamete from the heterozygous parent and the other from the homozygous recessive parent. As earlier discussed, the gametes produced by homozygous recessive parent all would be the same, and there would be 8 different types of gametes produced by the heterozygous parent, and hence the gamete of or the genotype of heterozygous parent would be expressed in the progeny. In the first case, where no crossing over takes place, the phenotype is capital ABC and small abc, and the hypothetical frequency assigned is small a and small b. In second case, when a single crossover takes place between the gene A and B, the phenotype observed in the offspring is capital A, small b and small c, small a, capital B and capital C. The frequency assigned is small c and small d. Then the third case, where a single crossing over takes place between the gene B and C, the observed phenotype in the progeny is capital A, capital B and small c, small a, small b and capital C. The supposed observed frequency assigned is small e and small f. Then the fourth and the last case, that is double crossing over. Here crossing over takes between gene A and B and gene B and C. Here the observed phenotype in the offspring is capital A, small b, capital C, and small a, capital B and small c. The number of offspring of frequency to be observed is assigned as small g and small h here. The total number of all these frequency or the sample size is capital T. If we consider the recombination value or crossing over between A and B as X, crossing over between B and C as Y, and A and C as Z, then X would be equal to the total of frequencies associated with the crossing over between A and B gene, that is C plus D plus G plus H upon the total sample size, that is total of frequencies into 100. Similarly, the recombination value Y% would be equal to the total of frequencies associated with recombination between B and C gene, that is E plus F plus G plus H upon the total sample size, that is total of frequencies into 100. And Z% would be C plus D plus E plus F upon total frequency into 100. To find the order or sequence of genes, if this value Z is equal to X plus Y, the sequence of genes would be A, B and C. If Z is equal to X minus Y, the order would be A, C and B. If Z is Y minus X, then the order of genes would be B, A and C. We can understand this by making a linkage map. As here we can see, we have the genes in sequence A, B and C, and here Z is equal to X plus Y. This is the first case. In second case, the sequence of genes is A, C and B, and here if we subtract Y from X, we will get the value of Z. So Z is equal to X minus Y. And lastly, the third case where Z is equal to Y minus X, the sequence is B, A and C, and if we subtract X from Y, we will get the value of Z. So based on the data calculated above, we can easily make linkage maps. Here the unit used is map unit, and one map unit is equal to 1% recombination or crossing over. This unit is also referred to as H.Morgan in the honor of the scientist H.Morgan.